Welcome to this episode of Lectures with Liddington. Last episode, or last unit, we finished up talking about DNA and what that structure looks like and how we can make a copy. That leads into where we're going next, which is protein synthesis, making proteins, right? We talked about the fact that we use DNA in order to make proteins to give us our traits. In order to do that, we need this really important molecule called RNA. So let's talk about RNA. So RNA, what does it look like? Well, it's similar to DNA in the sense that it's another polynucleotide. Remember, deoxyribonucleic acid was DNA. Well, RNA also stands for something very similar, which is ribonucleic acid, and that has to do with the sugar that's involved in this backbone. So we've got the sugar phosphate backbone. It's ribose sugar there instead. But it's similar in the sense that it's a polynucleotide, so it's a big molecule that has many nucleotides. Those nucleotides are the monomers. There are many nucleotides attached, but it's only a single strand, and the bases are a little bit different. So we have A, U, C, and G. So the U is for uracil. Instead, we don't have thymine, we don't have T's in RNA. So this will become important as we go on. But for right now, just knowing that there's a replacement here, anywhere that you would expect a T to be, there's going to be a U, which is uracil. So let's put it right together, compare and contrast DNA to RNA. So these two molecules, these are our polynucleotides, and they're similar structures. So they have this twisted nature to them. They're made up of nucleotides. Nucleotides are the monomers, but they're a little different. So remember our DNA, it's a double helix. So it's double-stranded. It's two sides. It's a whole ladder that gets twisted. We have A, T, C, and G. So those are our bonding rules, A to T and C to G. And it's huge. This is a really big molecule. You have trillions of base pairs long in a DNA molecule. So it's really big. And we take that and we protect it in the nucleus of our cell. RNA, so single strand, so it's only that one. We have A, U, C, and G. So it would be A to U and C to G when we have to match things up. And it's not as big. So it's many base pairs long. So it's still a polynucleotide, but we don't have trillions of bases long. It's just little short segments. And the reason being is because we use RNA to help move the message. So DNA is really huge and it's stuck in the nucleus of that cell. Well, RNA is only a single strand and it's a little segment. So what it can do is it will come and bond with that DNA. So it can take that message and send it out of the nucleus, right? We don't want all of our proteins chilling inside the nucleus there. They need to go out. They need to get sent out of the cell in two different places. So that RNA is little, so it can fit out of the nucleus. Perfect. Single-stranded, smaller section, and it will travel out of the nucleus into the cytoplasm wherever it's really needed, depending on what it's doing. So there are 3 different types of RNA, and they all have slightly different jobs. They have that same structure. They're single-stranded, A, U's, C's, and G's, and they're little segments, but they all have slightly different jobs. So messenger RNA, this starts in the nucleus. This is the piece that is carrying the message from the DNA out of the nucleus. Then we have rRNA. This is ribosomal RNA, and this is really just structural. So this is what makes up our ribosomes. And then we have another molecule called transfer RNA or tRNA, and this is the piece of RNA. This is what makes those protein chains. So this one will attach amino acids to make those protein chains. So let's pause there and do a little practice.